Hi everyone. So welcome to supplementary video 6. And in this video, what essentially I wanted to do was essentially cover two examples of real world problems and show how uh, w- we can actually have real world problems and how we can reduce them to study 1D problems and make conclusions based on analysis. So this particular problem, which I'll first discuss, is also covered in the book. It's problem 3.4-3, and I'll describe this in a bit of detail. But this is an actual experimental setup, and this is a very famous group, basically, which works on biological systems and how transport phenomena can affect basically transport property can affect properties of biological system. So let's just read the title of this paper, and this is a 2006 paper, so not too old. And it says evolution of oxygen glucose concentration profiles in tissue mimetic cell culture system of embryonic stem cells. So what basically this paper is doing is essentially, if you see here, there's a bottom plate here which has these cells, right? And there is a region which is filled with the media. Okay, so there is a cell and it is filled with the media, and from the bottom portion, which is the layer exposed to the cells, you are supplying oxygen, and then they have basically placed these slides basically on the side, on the top, which block any oxygen to reach from the sides. But the sides which are open, which you see here, and the other side, there you can actually expect that the oxygen concentration is available. So there is some consumption of oxygen from the bottom layer by cells. Then there is oxygen present on the sides, and the top and these other side mediums are basically blocked, and these are called spacers. So they have blocked any flux of oxygen coming through the sides. So the idea behind this paper was essentially understanding how the transport, how the oxygen concentration will evolve within the system, because some cells are actually getting exposed more to the oxygen, and some are getting less exposed to the oxygen. Because you can imagine in this figure, in the y direction, because there are blockers in the y direction, the oxygen won't be same for all the cells. So this was essentially the problem setup. Now, naturally, there's a lot going on in this setup, but we can actually simplify the system and can actually analyze it with what we have already discussed in our class till now. So what I will do is I will just formulate the problem, and then I will leave it up to you to solve this problem. Okay. So let's go to the system. So this is the same setup which we saw last time in the previous slide. And what you see here in this bottom uh, layer here, which was basically z equal to 0, this was the layer of cells. And the assumption is that the cells are so thin, the cell layer is so thin, that it's basically there is no height of that layer. And then it was told to us that the surfaces I'll mark with yellow now, the top surfaces, and the side surfaces are actually all blocking surfaces. So they're blocking. So these surfaces are blocking oxygen flux. Okay, so again, just to repeat, we have a mammalian cell layer in the bottom, and then we have these top and the two sides where they're blocking, and then this side, which I hope becomes clear, this surface in the x direction in this picture, this one and this one behind in this direction, they're basically constant oxygen, constant oxygen. And that's because they have been exposed to air outside. So now in this problem, you can also see when do we use constant surface, constant flux boundary condition in actual real systems. Okay, so it is used when we have, when we have a reservoir. So this cell culture here essentially is exposed to oxygen outside. So the sides which are open will have a constant concentration. Now when we have blocked the surfaces from the top and the side, that's where we have constant flux and equal to 0. And then there is only one surface left, right? So there are 5 surfaces we have discussed till now. Two surfaces, this one, basically this one, and this one, the one which is perpendicular to x, they're both at constant oxygen. The ones on the side are constant flux equal to 0. And what happens at the bottom? And that's where the assumption comes in, that actually oxygen is consumed by cells as a 0th order reaction. Okay, so oxygen, this is an assumption that the paper makes, assumption, and it's a pretty reasonable assumption that oxygen is consumed by cells as a 0th order reaction, as a 0th order reaction. Okay, and they call basically R of oxygen at the surface at z equal to 0 is essentially simply minus k naught, which is the rate, 0th order reaction rate. And note that this is a surface reaction. Okay, so this is also a surface reaction. This is not happening at the whole region, whole medium, whole cell culture. It is only happening at the bottom layer where the cells are present. The remaining, it's just diffusion, and there is no convection, and yeah, their assumptions are no convection and steady. Assumptions, further assumptions, no convection and steady. No convection and steady. So essentially what we have to find is concentration of oxygen. So I'll say C is the concentration, CO2 is what we have to find. So I'll not write O2, I'll just write Ca just to make it simple. Okay, A is the concentration of O2. So this is what we have to find. Now this is a steady system. This has been given to us. And what I want to do now is write the governing equations and boundary conditions. And then I'll give a solution strategy, which I hope you will all try yourself. So this is a CA of X, comma Y, comma Z, right? So we have 3 dimensions. Now, we know that the surfaces in the Y direction here, which is this one and this one here, the 2 surfaces, they're actually not accepting any flux. So there is no flux here, right? There is all yellow surfaces, but both the surfaces in the Y direction, they're not accepting any flux. And if you have not seen the video supplementary video 5, I invite you to go and see that video. Is that if there is no flux in that direction and there is symmetry, there is no reason for concentration to depend on Y, because nothing is changing in the Y direction. The flux is 0 there, the flux is 0 here. It's a steady system, so there can be no accumulation. So in minus out plus generated is equal to 0. There is no generation, right? There is no accumulation, because there is essentially a steady state. So in is equal to out, and in and out are both 0 itself. So everything is basically not dependent on Y. So you can actually drop this Y dependence. So you can drop this Y dependence. And this is an important point. And again, if you're confused, I invite you to check out supplementary video 5, where we discuss this in scenario 2 of why you can drop a dependence on one dimension if there is no flux in that direction. Okay. So if you accept that, that means Ca becomes X comma Z. Okay. So if Ca becomes X comma Z, what I can simply do is I can sketch this system and remove the Y plane. So I can essentially, I can look at this on the XZ plane. Okay. So let's do that. So how would we basically get the Z plane? So X is this direction and Z is the direction above. Okay. So let's look at the XZ plane. So at Z equal to 0, we have Z equal to 0, which was this plane, this plane here. Z equal to 0, we have cells. So this is cells here. Then we have a top plane at Z equal to the top plane. We have again a blocking slide. So we have a top is blocked, or flux is 0. And the sides, which are X is equal to, so now X is this side. So the surface I'll mark now, let me change the color. This this one, the red one, the red surface is constant O2, the one which is essentially, so let me not change the color. The red marked arrow which says constant O2 is the one in the X direction. So this is essentially constant O2. Constant O2. and here is constant O2. Okay, so this is Z, this is X. Okay, and then this is constant O2. So the paper uses this to be H, and is it 2W or it's W? This whole thing is W. Okay, so this is now our system X and Z, and we have the equation. So first thing is governing equation. And note again that there is no volumetric reaction. So governing equation, so it's del square Ca by del x square plus del square Ca by del z square is equal to 0. And we don't need to find any dependence on y, because there is no flux in y and there is no reason for it to have any dependence on y. Okay, so this is our governing equation, and what we want to do is now write the boundary conditions. So let's write the boundary conditions. And boundary conditions, as we have discussed, will be two boundary conditions in x, because there is x square in the denominator, and two boundary conditions in z. Okay, so we know that we can simply say that Ca at x is equal to plus minus w by 2 is C naught, something which we have been given, basically the constant concentration, constant O2, right? And experimentally also, you can imagine this is very easy to know. The other option is you can also take you can simply say that there is symmetry about x is equal to 0. Both are okay, right? But I just use this one. You can use the symmetry one too, and then keep one as the constant concentration. Both are okay. And then the concentration we want to find in Z. So in Z, we know that cells are consuming oxygen on this layer at Z equal to 0. And then the top layer is blocked. So top is easy because if it is blocked, the flux is 0, which means that del Ca by del Z at Z equal to H is actually simply equal to 0. Now the other condition is what is happening at the cell site. And that's where basically we have to use an interfacial balance. So what is happening is oxygen is being consumed by the cells, and then it is being transported up. So simply, you can imagine that this is the cell layer, and below the cell layer there is basically no flux, right? So this is phase one. This is phase one. This is phase two, and n hat is Ey. Right? So I'm doing an interfacial balance. So I'm doing an interfacial balance. And if you once you do this, basically what you say is F1, and there is no velocity of the interface. So F1 minus F1z, because n hat is Ez, not Ey, mistake, Ez here. F1z minus F2z plus Bs is equal to 0, right? This is our interfacial balance, that the flux coming in minus going out plus generated is equal to 0, because there is no accumulation. And F1z is basically 0. F2z is minus of minus d del Ca by del z. And Bs is the rate of generation, or minus k naught, which we have discussed, minus k naught to 0, or del Ca by del z at z equal to 0 is k naught. K naught. So there are four boundary conditions that we did. One is plus minus w by 2, one is z equal to h, and one is this one. Okay. Now, so we started with a 3D problem, we had done a 2D problem, and now you can further make it a 1D problem. And remember that we have discussed this in supplementary video 5, is when you have a constant flux in both the z limits, when you have a constant flux, you can integrate the equation by defining an average concentration, which will be here in this case 0 to h Ca dz by h. Okay, so I'm not repeating myself here, but essentially what I'm trying to show you here is that it is possible in a real world system to start from a 3D model, 3D picture. Because of the no flux in y, we can remove that term, then we can write boundary conditions, and because of this nature of constant flux, we can actually make it a 1D problem and solve it analytically. And that was done in the paper, and there are you should definitely check it out if you're interested. And this is also in the book example 3.4-3 if you want to take a look. Okay. Now note one thing which I did not basically I want to emphasize is that these boundary conditions, constant flux, constant concentration, and interfacial balance, all three were used in this problem, and this is a real system. So when you use a constant concentration, that basically means that you have, excess, you have a reservoir. So in this case, it was basically the cell culture was available, the air was available. Then we had these blockages, which was basically making the constant flux to be zero. So constant concentration was a reservoir kind of condition. Constant flux was, or constant flux equal to zero in this case was blockades. And then we have the cell media, where we assume because they are being consumed by the zeroth order reaction, it became like an interfacial balance where you have the flux going on and is equal to being consumed. So in this way, in a real world example, you can see all three kinds of boundary conditions are present, yet because of the simplicity of the system and the experimental design, you can actually make a simple model that is able to explain the experimental data and provide you insights. So I hope this video was useful. Before I go, I want to just quickly mention one another paper, and I won't be writing down the equations or anything here, but this paper actually is about microfluidics. So I imagine some of you will be doing experiments or collaborating with experimentalists who will be doing microfluidics. And what you see here essentially is set up a microfluidic system where the bottom slide here, if you look at this, the where it says glass slide, what you're doing is you're filling this white chamber, which you see in here, white chamber, with some sort of a polymer, which you're trying to polymerize using UV. Okay. And what these authors noted was that sometimes because this structure, typically the outside layer, the PDMS device, which is noted here, has oxygen that diffuses through the PDMS, that can inhibit your polymerization and create these layers here where you will not have a polymerization. So what these authors did was, they took a 1D model, simple 1D model. They said we'll ignore the other directions. What we are interested in is finding what is happening on top here and bottom here. So they only modeled the Z direction, did a reaction, diffusion, unsteady reaction, diffusion system, and solved it and matched with the experiments. So again, this was a real world system where you had oxygen diffusing in through a PDMS device, which was inhibiting your polymerization. So as you can see, oxygen inhibited uh, polymerization in the title, and that they modeled it and also matched with experimental data. So again, what we are discussing now in the class is actually applicable to real world systems. And you can use the same things: constant boundary conditions, constant flux conditions. And I imagine a lot of you will be doing similar experiments and models in your PhD. So you can keep this in mind, that these systems, 1D model study systems, are not just ideal systems. They can also actually exist in real world. Okay, so with that, I'll close this video and I look forward to seeing you in the next video.